So I think, Sumati, with that, we'll ask you to kick off, if that's all right.
Thank you, David, and good morning, everybody. Thank you for coming. It's early in the morning, and it's good to see that at least a handful of people interested in understanding the regulatory considerations. So I think what we're going to do is maybe in the next 10 minutes or so, I'll give you a very high-level overview of some important concepts that are relevant to antifungal drug development. And then after Radu's presentation, we have a series of questions that the moderators have for us. And I think this background will be very helpful in addressing those questions because it's essentially a lot of these same topics that will come back. So it's good that in the last few years we are seeing some increased interest in developing antifungal drugs, and not quite to the extent we would like it, but at least there is some uptick. And I think it's important to keep in mind that many of the principles that underlie antifungal drug development are very similar to those for antibacterial drug development. And if you've been following that field in the last decade or so, there's been significant churn, but also a fair amount of progress that has been made. So here are some examples of indications that we have currently approved products for. Systemic infections like invasive aspergillosis, candidemia, mucormycosis, and the mucosal diseases such as esophageal candidiasis and vulvovaginal candidiasis. There's some interest in new indications, particularly infections due to other rare molds, allergic bronchopulmonary aspergillosis, and recurrent VVC, indications for which we don't have any approved products right now. And there's also some renewed interest in developing drugs for for diseases such as cryptococcal meningitis and more so for coccidioidomycosis. So just to make sure that everybody's on the same page, we really have two pathways for product approval in the United States. One is the standard approval, where the approval decision is really based on an endpoint, which is clinical. So it really assesses how a patient feels, functions, or survives. Then the other pathway is the accelerated approval pathway, which is based on a surrogate endpoint that is reasonably likely to predict clinical benefit, or it can be an intermediate clinical endpoint, which is measured earlier than irreversible morbidity or mortality. And I've given you the references if you need more information. For NDAs that have come to us for review, the data packages have generally included one trial per indication. So you can have a trial in invasive aspergillosis and a trial in invasive mucormycosis, the approach that was taken for the isovocconazonium NDA. Or it might be just one trial and good supportive information from non-clinical and in vitro studies. And the reason I have the bullet about orphan designation, because I think there is a misunderstanding that if there is orphan designation, which a lot of antifungal products have, that there isn't a need to provide adequate data from adequate and well-controlled trials. And that's really not true, because our statutory standard does not change. We do need effectiveness demonstrated in adequate, well-controlled investigations. Now, certainly for certain orphan indications, historical or external controls. Okay, so the trial designs that have been used for recent approvals have generally used a non-inferiority trial design, and it's easy to justify a non-inferiority margin for the severe invasive fungal infections. And also want to note that we have been willing to accept wider non-inferiority margins, which translates to smaller sample sizes for products that have the potential to address an unmet need. And placebo-controlled superiority trials are recommended for a VVC indication. Common endpoints have included all-cause mortality or clinical success, and it's usually at a fixed time point. It's 6 weeks or 12 weeks. I think it's very important just to spend a little time on discussing endpoints, because again it comes up in the context of the questions. I think it's very important that whatever endpoint we decide to pick for these clinical trials have to reflect some benefit for the patients. And as is outlined in our Code of Federal Regulations, I think it's very important that the method for assessment of the subject's response should be well defined and reliable. And this is sort of a recurrent theme that comes up. So as I've already mentioned, we accept a clinical endpoint that can measure how a patient feels, functions, or survives, or in some instances, a surrogate mark endpoint. So here are some examples of endpoints that I've primarily borrowed from the antibacterial space. For a clinical endpoint, it's usually a clinical response. Sort of at a fixed time point, is a patient a success or a failure, or it might be a time to event analysis. All-cause mortality, which I've mentioned, we've used for invasive aspergillosis. It's not very common that we recommend a microbiologic response as a primary endpoint, but there are conditions like, say, a gonococcal infection, where a microbiologic response is acceptable as the primary endpoint. For conditions like complicated urinary tract infections, we would do a combination, so clinical success and a microbiologic success. An example of surrogate endpoint in the anti-infective space would be sputum culture conversion for tuberculosis. We've also, about 2 or 3 years ago, approved benzonidazole for the treatment of Chagas disease in pediatric patients based on a surrogate endpoint of seroconversion. I won't go through this, it's really here for your reference, but the important point is that a surrogate's a biomarker, but every biomarker is not a surrogate, because for a biomarker to be used as a surrogate that's predictive of clinical outcome, we need information that changes in the biomarker actually correlate with changes in the clinical outcome. I've just given you this again for your reference. There are some resources available if you're interested in the biomarker qualification process that the FDA has. And in the table, you can see that galactomannan went through the process in the old system, but is a recognized biomarker. And then in the anti-infective space, the other one we recognized was for the malaria challenge studies, it's a PCR, Plasmodium 18 as PCR. So what would be the role of diagnostics in anti-infective trials in general? I think a couple of points. If you want to use a diagnostic test for enrichment purposes in the clinical trial, they don't necessarily have to be FDA cleared or FDA approved. If it's FDA cleared or approved, it's straightforward, but if it isn't, then we do ask for some information on the performance characteristics of the test. We work closely with colleagues in the Center for Devices and Radiologic Health. I think we have allowed, say, for candidemia and candidiasis trials, use of non-culture-based methods for enrollment purposes. We've allowed the use of galactomannan in trials of invasive aspergillosis. And just like with diagnostics, not having to be FDA cleared or FDA approved to be used in tests, I think in trials, qualification of an endpoint is also a not prerequisite if it needs to be used in clinical trials. I know there's some interest in the audience here about inhaled antifungal therapies. Again, it's a relatively new area. We don't have a lot of experience so far. But I think it's important to keep in mind that there have been development programs for, say, NTM infections or non-CF bronchiectasis, and there are important lessons that can be learned from those programs that I think have a bearing on developing inhaled antifungals as well. And as I've already mentioned, if this is an avenue you're interested in pursuing, I think it's really important to keep in mind that you select a clinically meaningful endpoint and not one that's based solely on a biomarker or a laboratory test. We'd also recommend that you do adequate development work, including phase 2 trials, because that will really help you in designing the phase 3 trials. And otherwise, you'd be in a situation where we've done phase 3 trials, and if things don't go quite well, then I think we've not done a favor to anybody. I think also important to keep in mind that for some of these disease conditions, there's a fair degree of heterogeneity in the patient population. And because of that, there can be a fair degree of variability in treatment effect. And so your trial may not be successful, not because the product doesn't work, but because of the patient population that was enrolled. So you sort of have to balance that with having an extremely restricted patient population, because then that affects the generalizability of the results. I think I also want to note that we in the division are really not pulmonologists. Most of us come from an infectious disease background, and a lot of these kinds of diseases, I think there's a fair degree of involvement with the pulmonologists. So we do work very closely with the experts in CEDAR in our center who have expertise in providing regulatory oversights for pulmonary products. There was some interest in learning, understanding the incentives that are available for antifungal drugs, and comes up in the questions. So I have a couple of slides, quickly go through them. One such incentive is the QiDP, or Qualified Infectious Disease Product Designation, which is available for antibacterial or antifungal human drugs that are intended to treat serious or life-threatening infections, which for the most part includes the vast majority of severe fungal infections. I think if a product has a QiDP designation, what it provides is additional 5 years of marketing exclusivity. And for the first application that's submitted to us, it's an automatic priority review, even if you don't necessarily meet the other criteria for priority review. And the product's also eligible for fast track designation. The other incentive might be of interest to this group is the Tropical Disease Priority Review Voucher, which is for products that are used to prevent or treat diseases which are listed under the Food, Drug and Cosmetic Act. And one of the fungal infections listed there is cryptococcal meningitis. And there are certain criteria that need to be met to be eligible for the PRV. And I think again, this is more for your reference, but the main point here is even though there is an existing list, there is a mechanism by which other conditions, other infections can be added, and there is a docket to which you can submit a request should you be interested. LPAD pathway, I'll just spend a minute on this. This is the limited population antibacterial antifungal pathway for antibacterial antifungal drugs. This is part of 21st Century Cures Act, which was signed into law in December 2016. And what this really provides is a little more flexibility for us in the risk-benefit assessment. I think really taking into consideration the severity, the rarity, and the prevalence of the infection. But again, very important to note that our standards for approval do not change. So even if it's a product that is being submitted for approval under the LPAD pathway, we do need substantial evidence of effectiveness. And there are some conditions if a product is approved under the LPAD with regard to labeling and promotional materials. So far, we have approved 2 drugs under the LPAD pathway, Arikayce, inhaled amikacin for non-tuberculous mycobacteria, and Pretominate for the treatment of XDR and MDR intolerant or non-responsive tuberculosis. Again, for both of these products, the approved patient population was very limited and very well defined and specific. We had treatment effect demonstrated in at least one adequate and well-controlled trial for each of the products, and we considered the benefit-risk profile to be acceptable in this very limited patient population who had very few treatment options or none. And the limitations of data reflected in product labeling. So I've just given you the 2 examples, and this is what the highlights look like. And we are required to have limited population written in specific places, and the text that I've highlighted is also required language. So before I stop, I'll just make a pitch for studying drugs in children as well. As you're aware, under the Pediatric Research Equity Act, pediatric studies are required unless the requirement is waived, deferred, or not applicable. At least in the United States, many antifungal drugs do receive orphan designation, and so they are exempt from this requirement. However, we do highly encourage sponsors who are developing antifungal drugs that they consider developing it for pediatric population as well, because children do need safe and effective therapies. For the most part, we are able to extrapolate efficacy from adults, so we're not looking for randomized controlled trials in the pediatric population. We need some information on safety. And I think as an incentive, we are willing to consider issuing a pediatric written request, which then gives you some period of exclusivity. So in summary, I've provided a high-level overview of the key considerations in antifungal drug development. Again, some of these concepts we'll discuss in further detail during the Q&A session. As I've mentioned, many of the principles that guide antifungal drug development are really very similar to those for antibacterial drug development. I've just provided for you some examples of incentives and pathways that are relevant to antifungal drug development. And really, if you are embarking on a program where we don't have a lot of prior experience, we highly recommend that you come talk to us. More than willing to engage with you multiple times so that at the end of the day, we have a development program that has a high likelihood of success and patients have new, safe, and effective therapies available. So again, thank you for coming, and my contact information is here should you have any questions. Thank you.
Thank you very much.